In this video, we will discuss whether this AMT, that is adipose manipulation transplantation, can suppress cancer progressions through other metabolic pathways. In the previous videos, we have discussed the suppression of cancer progressions through the manipulation in the adipose, in the glycolytic genes or fatty acid oxidation pathway. Here we will discuss if we disrupt these uridine based pathways, whether it can also suppress the cancer progressions or not. So in order to do that, they use this gene UPP1 genes, and the upregulation of these UPP1 genes will cause high uridine pathways requirement by the adipose cells compared to its control, where they use BP64 gene. So when they co-culture those adipose manipulations, when they co-cultured those uh, manipulated adipose with the tumor cells or the pancreatic cancer cells, they are able to see that in the case where tumor cells are co-cultured with adipose with UPP1 genes upregulations, the number of cells goes down compared to the control case. And when they supplement the 1 millimolar uridine, they are able to see that number of cells are comparable to the control case, and there is not much difference, indicating that it's the uridine pathways that get disrupted, and because of that the number of cells goes down when number of tumor cells goes down when they are co-cultured with the adipose. And they check the other byproducts also, such as the ATP productions, and they are able to see the ATP also goes down when the tumors are co-cultured with adipose with UPP1 genes upregulations. And the ATP, we know that, is the uh, lowest form of energy that is present within the cells, and if ATP goes down, indicating that cells have less energy to perform their functions, and that will cause the cancer suppressions. And these things are regained when uh, cell culture medium is supplemented with 1 millimolar uridine, indicating that the uridine could be the reason behind the decrease in the ATPs. And the other things that they also check, other byproduct they also check, that is NADH and lactate. Both of them goes down in the case where tumor cells are co-cultured with adipose with UPP1 gene subregulations. Once they have the expected results from the in vitro studies, they also did the studies in the mice model or in the in vivo studies to check whether these results are reproducible in vivo or not. So similar to their previous experimental model, that is, first they inject the PANC1 using xenograft model into the mice. After 4 weeks, once the tumor is developed, they transplant this UPP1 CRISPR-A adipose organoid. That is, this organoid has this UPP1 genes upregulations, and after 3 weeks they sacrifice the mice and check tumor volume, growth, hypoxia, angiogenesis. And what they are able to see, that in terms of the tumor volume, when they use when the tumors are mice is transplant with this UPP1 genes organoid, they are able to see a decrease in the tumors compared to the control case where tumors are transplant with the adipose in which UP64 gene is upregulated. And these things are statistically significant, that can be seen in the left hand side. Thereafter they check this proliferation markers, that is MKI67, and it also goes down, indicating that the cancer progressions goes down when the tumors are co-cultured with adipose with UPP1 genes is upregulated. And the other side product, that is NADH and lactate, these two also goes down in the case where tumors or mice with this PANC1 tumors is co-cultured with the adipose in which UPP1 genes are upregulated, compared to the case where tumors are co-cultured with the mice with pancreatic tumors are co-cultured with the organoid in which VP64 gene is upregulated. So together all these results indicates that their AMT approach can be used not just through this glycolytic or fatty acid pathways to suppress the cancer progression, but it can be used uh, on other metabolic pathways that can also cause suppressions of suppression of these cancer progressions by depriving those cells of those important nutrients that is necessary for the cancer progressions. So I hope you understood these results and how they come to the hypothesis that AMT can be used on different metabolic pathways to suppress the cancer progressions. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.